Hi all, welcome back to Microbiology Lectures. Last time out we talked about how basic controls of microbiology or microbe growth. Today we're going to kind of continue that, talk a little bit about the CDC's biosafety levels, just so you're informed. Like if you're entering a lab and it's a biosafety level of 1 or 2, what does that mean? You're likely not to enter a 3 or 4 zone without some knowledge prior to that. But just those kind of levels, and then we'll talk about the microbes, the highly resistant microbes, and kind of go from there. So the first thing, there are biosafety levels that are set forth by the Center of Disease and Control and Prevention, or the CDC, and they basically set up guidelines. And the reason why it's important for microbiology to know these guidelines or these levels is that treating the different organisms that you might encounter in a biosafety level 1 is very different than how you would treat the organisms in a biosafety level 3, for that matter. So just a little bit real quick, we'll go through it. So biosafety level 1, these are microbes that are often not known to cause disease in healthy people. And so a lot of college campuses, labs, they're a BSL 1. Some college campuses and labs might be a BSL 2, so they might work on both organisms that do cause disease but have limited potential for transmission because maybe they're incubated at a temperature outside of the temperature range of, say, a human. If we're incubating them far below the temperature of a human, then often that will limit how much potential for transmission, or there's lots of ways in which we can limit the transmission of these different organisms. For the most part, when it comes to BSL 1 and 2, the supplies, the PPE, the personal protective equipment that you need are very similar. Normally 2 requires a little bit more, but most laboratories they use the same PPE for both biosafety levels 1 and 2. Okay, a biosafety level 3 means that you are dealing with pathogens that can cause serious or potential fatal diseases, and so therefore biosafety level 3, you have to have special gear, special permission, a lot more procedures on who enters that workspace, and a lot more training when it comes to working with biosafety level 3 organisms. And of course, biosafety level 4 is even one step above that. These are the organisms that are typically transmitted, and result in fatality or virulence in the organism that is transmitted to. So they're deadly pathogens. Okay, so as far as equipment, most of you who might be watching this would be familiar with the BSL 1 and BSL 2 equipment, which would be a lab coat, a mask, and goggles, and rubber gloves, and closed-toed shoes, long pants, this kind of equipment, protecting your skin from abrasion, from microbes that might enter your skin, but not to the point where you need an external air source, right? And you don't need a full hood like a biosafety level 3 and 4. Okay? So again, normally when we're dealing with biosafety level 4, these are deadly pathogens. So They don't just cause disease, but they're virulent. They cause death of the organism. And so those biosafety level 4s, normally what would occur is, of course, you have an outside air source, a full bodysuit of equipment, and then you're also interacting with these microbes in a closed, secure place, not in an open room. You can utilize biosafety level 3. You could be in an open room and move things in an open airspace, but when we're talking biosafety level 4, it's in closed hoods with their own air system, and that's kind of one of the things that kind of separate these two. Okay, so that's just for your information. Most people won't be working on 4 and 3 without a lot more training in microbiology, but some of you may enter labs that deal with biosafety level 2, and just be prepared that those organisms could cause disease, but we have measures to try to limit the potential for that transmission, normally incubation temperature or something along that line. Okay. All right, so remember the goal of these lectures is to talk about controlling growth. We already talked about how microbes grow. Now we're talking about how do we go about controlling their growth. Well, the first kind of process of controlling growth is know what is growing. Okay, so you have to know the procedure that you're going to use to control it is going to typically be based on, well, what is growing. And that being said, even though last lecture I talked about sterilization being probably the best method, still it's not an ideal method. It's not the silver bullet. It is a method that works, and it causes it allows for equipment to be sterilized and pathogen free, but it definitely has its drawbacks on what can be sterilized and the process itself, and we'll talk about those drawbacks in a little bit. Okay, so again, what type of antimicrobial procedure are you going to use? Are you going to use sterilization, disinfection? What are you going to use? Are you going to use a chemical base? Are you going to use heat and pressure? Are you just going to boil the material? This will all depend on the type and number of microbes. Okay? And so are we dealing with viruses? Are we dealing with bacteria? Are we dealing with protozoans? Like what is the organism? And a big piece of this is, what is the composition of the item that you're trying to treat? Is it living tissue? Are you prepping for surgery, or is it a wound, or is it like utensils? Is it a desk? Is it the air in a room? Like all these things, it really determines what procedure you're going to need. Environmental conditions, so we'll talk a little bit about the role that heat plays, pH plays, and then of course the risk of infection. What level are we talking about? Is this a biosafety level 1, not likely to cause illness? Biosafety level 2, that this kind of information needs to come to the forefront. Okay. So let's look at some types of microbes, that these multiple high resistant microbes. And so I'm going to give you some examples from the different kingdoms that we might encounter or are typically encountered when looking at types of microbes that are highly resistant. Okay. The first one that we're always concerned with is bacteria that can produce endospores. Okay, we talked about the endospore production and how bacteria go about producing endospores. In extreme environments can entice the bacteria to go through the endosporation. and create these endospores. And so when they do that, the endospore itself, the purpose of the endospore for that species is to resist extreme environments. Okay, so they go through the process when the environment starts to change, like starts to heat up, or the water starts to evaporate off, or the salinity starts to increase. Whenever the environment starts to change, and in some cases when we spray them with, some kind of chemical, it can entice them to go into a endospore formation, and then that endospore is resistant of, sometimes antibiotics, sometimes disinfectants, heat, okay, pH, lots of different things. And so there are ways to test to see if the organism can produce endospores. Okay, so there are dyes that will dye endospores. You can see here, based on this kind of microscope picture, that you have these bacilli bacteria, and then these little bluish green globs there are endospores that were produced by the bacteria. Okay. Another group that often resist disinfectants are protozoan cysts and oocysts. Although they kind of resemble and kind of look and sometimes it may seem like they have the same function as an endospore, they're not the same as an endospore. So a protozoan cyst or oocyst is the reproductive structure for that organism. So for example, like a Giardia will produce a cyst, and that cyst is like an encapsulation of juvenile cells or a juvenile organism, and it gets defecated out, and then it will sit in a water source until it gets consumed again, or it doesn't have to be a water source, it can be soil also, until it's consumed again. And then when it reaches certain temperatures, that organism hatches, multiplies, reproduces, produces more cysts, and can cause diarrhea, vomiting, other kind of things. Okay, oocysts, similar to cysts, okay, but again, these are reproductive kind of entities. It's how the organism reproduces. It's not how the organism resists changes in environment. So the oocyst, cyst and oocyst are very similar, but they're normally named different things based on the organism. So like an oocyst is like Cryptosporidium produces oocyst. Malaria would produce oocyst. But similar function. Okay, Cryptosporidium, same kind of pathway that Giardia does. So organism defecates in a water system or soil, wet soil, and then that oocyst would hatch when it enters a warm organism's digestive tract and irritate that digestive tract, which the body would then try to rid itself of that infection, and so diarrhea or vomiting occurs. Okay, but from a protozoan perspective, the cyst and oocyst reproductive parts, this is how they pass their genetic material on to the next generation. This is how they get from point A to point B. We can boil water that would have oocysts and cysts in it, and that will destroy the cyst or the oocyst. What you can't often do is spray it with disinfectants. They're extremely resistant to most disinfectants, because the disinfectants are working on, a lot of the disinfectants are working on kind of destroying a lipid layer, and the oocyst, the outer part of the oocyst, resists that disinfectant. So the best method, I guess, would be to boil it. Okay, there's quite a few others. So Macrobacterium, probably the species that we're most familiar with, or we know like the most about, is tuberculosis. Okay, they have a waxy cell wall. okay, which makes them resistant to a lot of chemical treatments. And so not only do they have that peptoglycan layer, but they have a layer of lipid proteins that are associated with that peptoglycan layer. The lipid causes them to reject things that are hydrophilic. The proteins allow them to use efflux proteins or pump things back out. So when things enter, they can get pumped back out. So again, highly resistant to many chemical treatments. Pseudomonas species, also very resistant. Normally the resistance coming from Pseudomonas is the fact that they readily form biofilms, and so they attach to surfaces, form a biofilm, and then they can be fairly hard to get rid of because that biofilm often resists disinfectants and things like that. And then you have a whole group of viruses that lack lipid envelopes. Okay, so you have lots of viruses that do have lipid envelopes. Okay, so they have like an envelope that goes around the outside protein that has glycoproteins attached to it and whatnot. But you have these viruses that lack that envelope, and you would think, well, maybe they're easier to get rid of, but a lot of our disinfectants, because they're reacting to lipids or tend to break or cleave lipids and cause the material to leak out, or allows for things to enter and desiccate the living, or in this case, because we're talking about viruses, the non-living material. That's how a lot of our disinfectants work. In the case of things that don't have lipids, then it's not breaking the lipids, it's not deteriorating the lipids, and instead they just have this hard protein casing or protein capsule. And lucky for us, many times if you heat these quite a bit, you can get that outside protein casing to denature. So there are ways at which all of these things can be treated, but they show resistance to many different things. And so we'll talk about the process of choosing different methods to treat different resistant microbes. Sometimes it's 2 or 3 methods together, and that's the best agent to get rid of that microbe. Okay. Next time we'll talk about numbers of microbes and how we go about determining whether or not a new disinfectant is worth selling or worth using, especially when we talk about maybe some of the greener options, the plant-based options for disinfectants. And we have to have a way to rank these chemicals that are coming on the market, and so we normally do that by looking at a D value, which I'll come back and talk to you about later next time.